Can somebody confirm and hear me?
I can hear you.
Thank you. Let me share my screen. That's not where I want to be. Here we go. Should be seeing my screen now. Not much to see, but today is July 24th, and we should be on chapter 4. For the lab today, we'll cover lab 5, streaking for colony isolation, and we'll talk about culture media. Any questions about what we're going to do today? Excuse me. I've learned that I get the background showing nicely on my screen if I have the room really dark, which doesn't make any sense at all. But anyways, it's working well. All right, last time we had talked about flagella. Any questions about flagella? I do realize that if the bacteria or the archaebacteria can move, swim or move, it has a flagella because that's the only structure that bacteria have for moving. It's not true of eukaryote cells, but for bacteria, if they can swim or move, it's because they have at least one or more flagellum. You can classify the flagellum from their arrangement on the cell. Let me blow this slide up. So you have monotrichous, and that means there's one on one end of the flagella. Amphitrichous, there's one on each end of the, did I say the flagellum? I meant the cell. There's one on each end of the cell. Lophotrichous, there's multiple on each end of the cell. And then peritrichous, there's many of them all over the cell. Remember that when you see a flagellar arrangement in a species, it will have the highest order of flagellum it can have, and then every lower order down to zero, because when the cell divides, like this cell here, if it were to divide, it would have none until it makes one flagellum. So a monotrichous species can have cells with one flagellum or with zero flagellum. And then an amphitrichous species can have some cells which are amphitrichous, some cells which are monotrichous, and then some cells which have no flagellum. Any question about any of that? So if cells are motile, meaning they can swim, rotate, run, or tumble, they have at least one flagellum. If they can move, that's called taxis, the movement toward or away from a stimuli. Here we have a light source up here, and these cells are swimming away from the light. So that is phototaxis, meaning they um, are responding to the stimuli, in this case, light, photo. And these cells are not photosynthetic because they're moving away from the light. There's also chemotaxis. Those are cells which are stimulated from a chemical. either toward or away from the chemical stimulus. Any question about any of that? All right, let's talk about the endoflagella. There are some cells which have endoflagella, also called axial filaments, and these cells are only spirochetes. This is a flagella, which is anchored on one end of the cell and then wraps around the cell in this sort of a fashion. Let me blow this way up. And we note that this flagella is inside the cell wall. So the pink is the cell wall, and then the bright pink is the axial filaments. So the endoflagella, or is a flagellum which is inside the cell wall, running like that in the cell, and it gives the cell a unique corkscrew-like motion when it moves. See if I can get that going. Hopefully that's going to run. There it goes.
Now we got to go help people customize and save hundreds with Liberty Mutual.
After the commercial.
Libby, where's the golf cart?
As you can see, let's back that up a bit. The cell just rotates one way to move this way, rotates the other way to move that way. And there you can see the cell is so agile that it actually bent in half and now it's moving this way and that way. Any questions about any of that? And this one is just moving this way by rotating that way. Any questions about the corkscrew-like movement of the spirochetes having the endoflagella? Fimbriae are hair-like structures found on a cell. Oops, didn't want to be there. They're the yellow structures here. They're shorter than flagella. They're also shorter than sex pili. And there's usually multiple fimbriae on the cell. They are not used in locomotion, meaning they're not used for swimming. They are used for attaching to a surface. And so some bacteria that are good at attaching to medical equipment or something like that, they have fimbriae. Sorry, I didn't say that right. Fimbriae. And then some human pathogens have fimbriae, which attach to the human receptors on our cells. All right, the pili, also called the sex pili, is longer than a fimbriae. Let me blow this slide up. And it's made by one cell here, which is called an F plus cell. And the sex pilus goes out and finds an F minus cell, this cell here, and then literally skewers it. And then it acts like a winch to bring the two cells closer together. The two cells do not have to be totally close together. Oops, I thought I had it, but apparently I don't. But the sex pilus is hollow, and then a plasmid can come out of this cell and then move through the sex pilus and then enter the F minus cell. So the sex pilus is used to transfer DNA from one cell to another. Pili is plural, pilus is singular, and there's only one here, so the sex pilus. Any question about the sex pili. The next structure of the prokaryote cell I want to talk about is the cell wall. If the cell has a shape, it's because of the cell wall. There are some bacteria that lack a cell wall, and these species that lack the cell wall, they're in one family. Let's see, the Mycobacterium. No, not Mycobacterium, no. Drawing a blank on the name of that. Ugh. It'll come to me. Anyways, there's one family that lacks a cell wall, and these cells tend to be pleomorphic, taking on many different shapes. Most bacteria have a cell wall, and that's why most bacteria are monophyletic. They only have one bacteria shape. The cell wall is complex, a semi-rigid structure responsible for giving the cell its shape. The cell wall protects the cell and prevents osmotic lysis. If the cell lacks a cell wall, then water can come into the cell, and that will expand the cell membrane, and then more water can come in until the cell membrane eventually will pop, and that will kill the cell because the cytoplasm can then leak out of the cell. But if the cell has a cell wall, the cell membrane can only expand up to the size of the cell wall. And then the cell wall prevents the cell membrane from expanding any further. If the cell membrane cannot expand, there's no further room for water to come into the cell. So that will prevent water from entering the cell, and that will prevent osmotic lysis. Now, to have osmotic lysis, you have to have a cell where there's more water in the environment than is in the cell, and that way water will move into the cell. Unfortunately for bacteria, they're often found in environments where there's more water outside the cell than inside the cell, and so water tends to move into the cell. But the cell wall, if it's present, will protect the bacteria. The cell wall is located outside of the cell membrane, but it is actually considered the last part of the cell. Anything the cell makes that's external to the cell wall, like the capsule, is considered external to the cell. So the cell wall is the last structure of the cell. And it's kind of curious that the cell wall is the last structure because the cell membrane is considered the gateway controlling what comes into and outside, leaving the cell. So the cell membrane is the gatekeeper, but it's not the last part of the cell. The last part of the cell is the cell wall, which is external to the cell membrane. The cell wall can contribute to the pathogenicity of some species, like the Mycobacterium. Part of the reason why the Mycobacterium can be pathogenic to human cells is because of the cell wall of the Mycobacterium. The cell wall is mainly made up of peptidoglycan, meaning it's the main molecule in the cell wall of bacteria. Any question about any of that? Now, in archaebacteria, many of them, probably most of them, have a cell wall, but they do not have peptidoglycan in the cell wall. They have another molecule in their cell wall. So peptidoglycan is only found in bacteria, and it's only found in the cell wall also. Any question about any of that? All right, peptidoglycan is a polymer of a repeating disaccharide. The two saccharides are NAG and NAM. NAG stands for right here, N-acetylglucosamine. NAM stands for N-acetylmuramic acid. You may learn either name because probably NAGS means as much to you as N-acetylglucosamine. Anyways, the point is, let me come on over here. The peptidoglycan is arranged NAM, NAG, NAM, NAG, NAM, NAG, just in a row of them. And then there's multiple rows next to each other, multiple rows underneath this one, or if this is inside the cell wall, there would be multiple rows on top of it as well. The NAG and the NAM are in the row linked covalently one to another. So they have a chemical bond linking this together. But then the rows of them are linked by a short peptide. So this row of NAM and NAG, come on, mouse, is linked to this row of NAM and NAG by this short peptide. And then this row here is linked together to this row by this short peptide, which actually links to this peptide and that peptide, but it links these together so that the short peptides are actually linking the NAG and NAM rows together. NAG and NAM are glycans. Glycans are just sugars. And then it's linked together by the short peptides, and that gives the molecule its name, peptidoglycan. Glycan linked together with short peptides. Any question about any of that? Penicillin actually inhibits the lattice formation, linking the short peptides together in the NAG and NAM chains. So if penicillin were to get in a growing cell wall, before the cell wall makes this short peptide linking that row to this row, penicillin can get in right there and then prevent that polypeptide from forming. And that's how penicillin actually damages the cell. When you prevent the lattice formation from forming in the cell wall, meaning prevent the short peptides from forming on the cell wall, you'll have this cell wall with a gap in the cell wall. The cell wall right there won't be complete because of the penicillin G blocking this peptide from forming, and that'll make a gap in the cell wall. The cell membrane can protrude out of the gap, and then as water comes into the cell, this cell membrane will protrude further outside of the gap of the cell wall. And as more water moves into the cell membrane will expand until the cell membrane will eventually burst out of that gap in the cell wall. And that would kill the cell, by the way. That's actually how penicillin, the whole family, damages bacteria, and that is it prevents the lattice formation in the cell wall, causing gaps in the cell wall where the cell membrane can protrude out of, and then as water moves into the cell, that protrusion will expand until the cell membrane pops out of the gap in the cell wall here. Any question about any of that? Let me remind you that the gram-negative cell wall has more than one layer of peptidoglycan. It's something like 3 to 20 layers of peptidoglycan. And then the gram-positive cell wall has something like 30 to over 100 layers of peptidoglycan. Any question about any of that? This is actually the gram-positive cell wall, showing you the layers of peptidoglycan. It's more than three. We only have three layers here. Actually, if it had only three layers, that'd be a gram-negative cell wall. And it has a cell membrane, this yellow structure here, the lipid bilayer of the cell membrane, which does have other molecules in it, but the main one is peptidoglycan. But it does have carbohydrates and proteins in it, and there's a protein there and a protein there. And outside of that is a very thick layer of peptidoglycan, showing you multiple layers of peptidoglycan, something like 30 to over 100. The main molecule is peptidoglycan in the cell wall of a gram-positive bacteria cell. But there are some other molecules, namely teichoic acid, which actually helps link the peptidoglycan together because it runs through the peptidoglycan, linking this one with that one, much like the short polypeptides. And then lipoteichoic acid, which actually links the peptidoglycan layers together and then also links the cell membrane to the cell wall. The lipid portion of the teichoic acid is in the cell wall here. Any question about any of that? And that's the gram-positive cell wall. It's pretty simple with teichoic acid, lipoteichoic acid, and peptidoglycan being the main molecules, peptidoglycan being the number one main molecule. There are some other molecules in the cell wall, like a couple of carbohydrates and some proteins, but these are the main molecules in the cell wall of a gram-positive cell. The gram-negative, I got to talk a little bit more about the teichoic acid. The teichoic acid may help regulate the movement of cations. The cation may move down the teichoic acid and then move into the cell. But teichoic acid is not needed for the cation to move into the cell because a cation can move through anything that's water soluble. And the cell wall of the gram-positive cell wall is water soluble, meaning any molecule which is dissolved in water will go right through the cell wall. Now, to enter the cell, you also have to enter the cell membrane. And for molecules that the cell wants, it will make a transport protein to move that molecule across the cell membrane. So like for all of the amino acids that the cell wants, it'll make a transport protein to move it into the cell. And then for all of the sugars like glucose and fructose and what's another one? Lactose, it will make a transport protein which will move that molecule into the across the cell membrane. And there will be a separate transport protein for each molecule. That's not entirely true. There are some transport proteins that can move more than one molecule into the cell. But for all of the carbohydrates, as well as the amino acids, there's only one transport protein that moves each molecule across the cell membrane. And the only exception to that I know, or there's a few drugs we have which mimic the molecule that the transport protein is trying to move across the cell membrane. And so these transport proteins are very specific. For our purposes, we'll say that they only move one molecule across the cell membrane. And then the polysaccharide of the teichoic acid can help provide antigenic variation between different species of cells, like E. coli, the normal E. coli has the normal polysaccharides on it. I don't know what it is, but E. coli O111 and what is it, H127 or 125, something like that. That antigenic variation comes from the polysaccharides, and then O111 is actually a human pathogen that antigen of O111 makes the cell pathogenic. And this would be an E. coli that you do not want because instead of helping you digest your food in your gut, this one could cause gastroenteritis. And that's just sort of something slightly important, as the polysaccharide gives the antigenic variation for the bacteria cell. Any question about any of that? All right, that's the gram-positive cell wall. Let's talk about the gram-negative cell wall. It's a little more complex than the gram-positive bacteria. Once again, we have the cell membrane being the yellow lipid bilayer right here, shown here. And above that, we have the peptidoglycan layer, which is thinner than the gram-positive peptidoglycan layer. It's always more than one. It's something like 3 to 20 layers of peptidoglycan, but it's only showing you one here. And then outside of that, we have another lipid layer. It's actually a bilayer. This layer of the outer membrane of the cell wall, this lipid bilayer is called the outer membrane of the cell wall. This layer of that outer membrane is actually very similar to the cell membrane. The main molecule is phospholipid. This layer does have phospholipids. It's probably the number one molecule in this layer of the bilayer, but it does have other lipids, mainly lipid A, this blob here, and the lipid A is attached to the O polysaccharide, making this molecule a lipopolysaccharide. The O polysaccharide is the hair-like molecule coming out. The important part of this molecule is the lipid A, because lipid A is an endotoxin. Toxins are something we'll talk about a little bit later in this lab or in this class, but toxins are something which are toxic to us. Any question about that? So lipid A is toxic to us. And if ever you have an infection by a gram-negative cell that has a cell wall, you will be exposed to lipid A, which is a toxin. Any question about any of that? So a patient who has a gram-negative infection, if that gram-negative bacteria has a cell wall, they'll always be exposed to lipid A. It is part of the outer membrane of the cell wall of the gram-negative cell, which they're the only ones that have the outer membrane of the cell wall. There are some other molecules in the outer membrane of the cell wall. There are carbohydrates and lipids in it. The main and proteins. The main one is that we're going to talk about is the porin protein. The porin protein is a channel protein that allows molecules to enter or leave the outer membrane of the cell wall. For example, the amino acid lysine is going to be wanted by this cell, and that lysine will move right through the porin protein to get through the outer membrane of the cell wall. It then will enter a transport protein in the cell membrane to enter the cell. Now, the carbohydrate glucose can also enter this porin protein, and then it will need a separate transport protein to bring in the glucose into the across the cell membrane. So the cell membrane is very specific, and we have one transport protein essentially to move each molecule across the cell membrane. So the cell membrane has many different transport proteins, each of them moving only one molecule across the cell membrane. But this porin protein tends to bring almost all of the molecules the cell wants through the porin protein. So it's a channel protein, allowing many different molecules to enter the outer membrane of the cell wall. But it does restrict some molecules from entering the outer membrane, like penicillin G cannot get through the porin protein. And penicillin G cannot damage the gram-negative cell because the porin protein does not let penicillin G enter the outer membrane of the cell wall. And remember, penicillin G has to get in the peptidoglycan layer to damage the cell. And so penicillin G does not damage gram-negative cells. Any question about any of that? All right. You don't really need to know much about the periplasm. That's the space of the cell wall containing the peptidoglycan between the cell membrane and the outer membrane. That's the periplasm. You should know that the outer membrane of the cell wall is a part of the cell wall. It's the outermost layer of the gram-negative cell wall. We do have some other molecules inside the gram-negative cell wall. There's the lipoprotein, which actually runs through the outer membrane and the cell wall. It may also combine into the cell membrane. There are proteins, mainly the main one is the porin protein in the cell wall. Anything else? And then do realize lipid A is the important molecule in the gram-negative cell wall because it is an endotoxin. I think I've already talked about all of these molecules. Yep, all right. So the gram-negative outer membrane is a little different. It does have some other molecules. The gram-negative cell wall, the gram-positive cell wall lacks. It does have lipopolysaccharides. That will be the where the endotoxin is, lipid A portion of this lipopolysaccharide. It does have lipoproteins and phospholipids in the cell wall. Phospholipids are absent in the gram-positive cell wall. The gram-negative outer membrane of the cell wall can help protect the cell from complement protein. That's a protein our body makes to defend the body from bacterial pathogens as well as other pathogens. And it cannot get through the outer membrane of the gram-negative cell, as well as antibodies. Some antibodies can't get through the outer membrane of the cell wall. And then it does protect the cell, the gram-negative cell, from penicillin G. So certain antibiotics are protected by the outer membrane of the cell wall. Now, do realize there are antibiotics that do go through the outer membrane of the cell wall. Let me go to the previous one here. And that is ampicillin, the third penicillin member of the penicillin family, as well as carboxyllin, does allow that member of the penicillin family to enter the porin protein. So ampicillin can get through the porin protein and then damage the cell wall. And I did already mention this, that the O-polysaccharide portion of the lipopolysaccharide can give antigenic variation for the cell, And here's one E. coli, which is a human pathogen, 0157H7. I think that's different than the one I said earlier. I think I said 0111. Both of those are human pathogens. So you don't want this in your gut because it can cause gastroenteritis. And that's kind of interesting. We always have E. coli in our gut. So that means in our gut we have lipid A, but this is a normal portion, normal, what do you call that? A normal part of our gut. So the gut is able to handle the endotoxin. And as long as the endotoxin is kept in the bacteria cell, it doesn't harm the cell. But obviously, that E. coli would eventually die. And if it dies in the gut, then that would release the endotoxin. And the gut cells would have to deal with that. I never thought about that before because our E. coli is in our gut as well as many other gram-negative bacteria. And that means our gut cells have to be exposed to endotoxin when the bacteria, gram-negative bacteria, dies in the gut. I already talked about the porin proteins. They're the channels through the outer membrane of the cell wall. So comparing the gram-positive cell wall to the gram-negative cell wall, the gram-positive cell wall have a thick peptidoglycan structure, giving the cell a rigid structure. The gram-negative cell walls have a thinner peptidoglycan layer. It does protect the cell from like osmotic lysis, but the cell is not as rigid. It is rigid in that all the cells will have the same shape, but it is not as rigid as the gram-positive cell wall. Teichoic acid is found in the gram-positive cell wall. There's no teichoic acid in the gram-negative cell wall. And then there's no outer membrane in the gram-positive cell wall. There is an outer membrane in the gram-negative cell wall. And inside that outer membrane is lipid A, which is an endotoxin. Any question about any of that? All right, the Gram stain mechanism happens by the cell wall. Crystal violet iodine complex forms in all cells. In the gram-positive cell, the crystal violet iodine complex does not leave the thick peptidoglycan layer. The decolorizing step does not remove the crystal violet iodine crystals. But in the gram-negative cell, the crystal violet iodine complex does wash out of the thinner peptidoglycan layer. The alcohol also dissolves the outer membrane because alcohol dissolves the lipids of the outer membrane. And then the cell stains pink when we stain it with safranin. Any question about the Gram stain mechanism?
I had a question.
Uh-huh.
So when in the gram stain, for the sorry, I'm forgetting. Can you go back to that last slide real quick?
Sure.
So for the gram positive, the CVL, does it
What's the CVL? You mean the
or not
CVI, the
yeah, the
crystal violet
CVI, sorry.
iodine complex?
Yeah.
Okay.
In the gram positive, is it still purple because it doesn't like wash out?
Yeah, and the gram-positive cells, the cells are purple. As long as you do the gram stain correctly, the cells are purple once you apply the crystal violet, and then they remain purple throughout the gram stain.
Okay, so
Although they
it
do
doesn't
become
go
a purple
colorless
purple
like the
when you add the
negative.
iodine. Sorry, what are you saying?
So it doesn't go colorless like the gram negative. It's just purple and not colorless in the middle.
Correct.
Okay, thank you.
All right. Actually, let's go next. So here's looking at the gram-positive cell wall compared to the gram-negative cell wall. You can ignore the lower portion of this table. The gram reaction The gram-positive retain the crystal violet iodine complex, keeping the cell purple, and the decolorizing step does remove the crystal violet iodine complex. It has a thick peptidoglycan layer, and the gram-negative is a thinner peptidoglycan layer. It's not single layer. This is a mistake in this table. It's about 3 to 20 layers of peptidoglycan. Teichoic acid is present in the gram-positive cell, absent in the gram-negative cell. You don't need to know about the periplasmic space. Obviously, it's only in the gram-negative cell, but the outer membrane is absent in the gram-positive cell, present in the gram-negative cell. And then lipopolysaccharide is none in the gram-positive cell, or virtually none, and then very high in the gram-negative cell. And remember, this is where the lipid A is, and so that's the endotoxin. All right, so that's the normal bacterial cell wall. There are some cells which are a little different, and we call them atypical cell walls. Any question about any of that? The first atypical cell wall is the archaea. These are prokaryote cells that belong to a different domain, and they do not have peptidoglycan in their cell wall. If the archaea have a cell wall, and most of them do, they have pseudomurine in their cell wall, not peptidoglycan. Pseudomurine is just another disaccharide. You don't really need to know about pseudomurine, but you should know the archaea have different cell walls lacking peptidoglycan. Any question about any of that? And then there are some bacteria that have atypical cell walls also. The Mycoplasmas is a family of bacteria that lack cell walls. So they don't have a cell wall. Obviously, the Mycoplasma has to be in a isotonic environment, because if they were in a environment with more water around than the cell has, the water will move into the cell, and the cell doesn't have a cell wall, and that would kill the Mycoplasma. So they tend to grow in isotonic solutions. The Mycoplasmas, interestingly, have a sterol in their plasma membrane, which gives strength and support to the plasma membrane. Now, if you remember, I did mention that animal cells also have a sterol in their plasma membrane to help give the cell membrane strength and support. And in animal cells, that sterol, we call it cholesterol. And if you want to avoid a diet containing cholesterol, you don't want to eat animal products, because cholesterol is only found in animal cells or animal tissue. So if you eat a plant diet, or I guess a bacterial diet, you won't get exposed to cholesterol. Mycoplasmas do not have cholesterol. It's another sterol, but it acts the same in the plasma membrane to give strength and support to the plasma membrane. Any question about the mycoplasmas? All right, there is another bacteria which has an atypical cell wall. It is the Mycobacterium. This bacteria is different because it has a lot of mycolic acid in the cell wall. And the mycolic acid gives this Mycobacterium different properties. Mycolic acid is a lipid, and there's so much mycolic acid in the cell wall that water-soluble molecules cannot easily move through the cell wall. And that includes many antibiotics have trouble killing Mycobacterium because most antibiotics are water-soluble. as well as bleach, which is water soluble. The mycobacterium would be resistant to bleach and most antibiotics that are water soluble. Now, that doesn't mean it's totally resistant to all antibiotics, because streptomycin is a lipid soluble antibiotic, and it goes right through the mycolic acid of the mycobacterium cell wall. Any question about any of that? All right, so the mycobacterium is the only bacteria that has a cell wall. You need to know it's different. It is true Nocardia also has some mycolic acid in it, but it's less than Mycobacterium. So Mycobacterium is the one to know. And then the other one is a bacteria that lacks a cell wall, and that's the Mycoplasma. And obviously that's atypical because it doesn't have a cell wall. Now, if the Mycoplasma doesn't have a cell wall, what's it going to stain with the gram stain? Is it going to be gram positive or gram negative?
Gram positive?
That's a good guess. You want to guess again?
Gram negative?
Yeah, it's gram negative. The crystal violet iodine complex will initially stain the cells, but there's no cell wall, no peptidoglycan to retain the crystal violet iodine complex, so it will wash out. So the mycoplasmas do stain gram negative because they don't have a cell wall, and so the crystal violet iodine complex does wash out. Any question about any of that? All right. You can damage the prokaryote cell wall. You don't need to know the names of the cells once you damage them, but you could damage the cell wall with penicillin, like penicillin G can damage the cell wall of a gram-positive cell, and then ampicillin can damage the cell wall of both gram-positive and gram-negative cell walls. So penicillin can damage the cell wall, preventing the peptide bridges from forming in the cell wall, and that'll make a gap in the cell wall, and then the cell membrane can protrude out of that gap. And then if water moves into the cell, that cell membrane will just protrude out of that gap in the cell wall further until the water eventually will pop the cell membrane. And then another way to damage the cell wall is to expose it to lysozyme. This is an enzyme that digests the disaccharide and the peptidoglycan. And so if you use lysozyme, you can actually make the cell have no cell wall. You don't need to know the term of it, but you can have the bacteria cell without a cell wall if you expose it to the enzyme lysozyme. And then that cell would be susceptible to osmotic lysis because if water moves into the cell membrane will just expand until it eventually pops. Any question about any of that? So I have mentioned already, gram-negative bacteria are not as susceptible to gram-positive bacteria to penicillin G because the channel protein prevents the penicillin G from getting across the outer membrane of the gram-negative cell wall, and then that will prevent the penicillin G from damaging the gram-negative cell. And penicillin G can get right through the cell wall of the gram-positive cell, and so it will damage the gram-positive cell. Now, that doesn't mean that there aren't antibiotics that can't damage the cell wall of a gram-negative cell. As I already mentioned, ampicillin can get through the outer membrane of the cell wall of a gram-negative cell. And so ampicillin can damage the cell wall of a gram-negative cell, as well as it can damage the cell wall of a gram-positive cell. And there are other antibiotics which can get through the gram-negative cell wall, like streptomycin is a lipid-soluble or fat-soluble antibiotic and can go right through the outer membrane of the gram-negative cell wall. All right, any questions about how to damage a prokaryote cell wall? If not, let's change gear and talk a little bit about the plasma membrane, also called the cell membrane. It is a phospholipid bilayer. Let me blow this slide up, which has two layers of phospholipids with the phosphate head facing outside the cell, because think of a red blood cell in the blood. The blood is mostly water, and you want the head, the phosphate group, to be facing the water. And then the head of this layer also faces inside the cell because the cytoplasm is mostly water. And then the tail portion of the phospholipid, the fatty acid portion, likes to be together around the lipid portions. So the tails come together because they like to be around lipid-soluble molecules, making the phospholipid bilayer of the cell membrane. The phospholipid is the main molecule in the cell membrane, or all membranes, really. We do have other membranes in the cell membrane, though, including peripheral proteins. That's a protein that spans the membrane. Peripheral protein is a membrane which is mainly on one side of the membrane. An integral protein and a transmembrane proteins, these are proteins that span the membrane. Like there's an integral protein, and that looks like a transport protein, which has to span the membrane because it will bind to a molecule, one molecule, and then transport it inside or outside of the cell. But there are other integral proteins that do span the membrane as well. And then there's a peripheral protein mainly on the outside of the cell membrane. And there's a peripheral protein mainly on the inside of the protein. A peripheral protein can have a portion inside the membrane, like this portion here. But the main part of the protein is on this side of the membrane, so that's why it's called a peripheral protein. Most of the proteins which are spanning the membrane tend to move around in the cell membrane. So this protein can move around, can move from here to like there, for example. And that's because this membrane is free floating in the cell membrane, and it can move around. The peripheral protein, if it's anchored to something, will not move about in the cell membrane. And that's because it's anchored to something that doesn't allow this end of the protein to move. And so it stays in one position in the cell membrane. And this protein here, if it's inside the cell, if it's anchored, it'll probably be anchored to the cytoskeleton inside the cell. It's a cytoskeleton is a lattice of proteins running inside the skull, giving the cell sort of a backbone. Anyways, if the peripheral protein is bound to the cytoskeleton, then this protein won't move. This peripheral protein, if it's bound to the extracellular matrix, this protein won't move in the cell membrane as well. Any question about any of that? So, I've obviously stated that the proteins can move around in the membrane, and that's because the membrane is actually a fluid. The membrane itself can move around. These phospholipids, not only can they spin in this position here, but this one can move from this position to that position. And that is the cell membrane is a fluid. It's about as fluid as olive oil at room temperature. It's kind of hard to get your head around that my hand here is, because I'm an animal, only held together by the cell membrane. And yet that cell membrane is a fluid. Those of you who have had A and P know that some proteins have to move around in the cell membrane in order for them to function, like a receptor protein. They have to come together. Once they're bound to their ligand, they have to come together, and then they will be brought into the cell to give the signal. So that one receptor that is bound does not move into the cell and then send a signal to the cell. They come together, and then a whole bunch of them are brought into the cell as one signal. And you need multiple receptors to be bound to before the cell will get the signal. But the point is that these proteins, if they're not anchored to anything, they're free to move about in the cell membrane. And the phospholipids can rotate and move laterally around as well. That's actually why water can move into the cell. And that is, water cannot get through the lipid bilayer. This fatty acid portion of the, let's see if I can blow this up, of the lipid bilayer will keep water from moving across. But every once in a while, this molecule will move over here, allowing water to come into this position. And then the water cannot move through the lipid layer right here. And so the water just waits until this molecule here moves over, and then the water can take this position and then move into the cell. And so the water just waits in position here until the phospholipid down here moves, and then the water can move into the cell. That's not a great way for water to move into the cell, but I'm just stating that water can move into a cell. For roots, water moves into the cell because there are transport proteins called aquaporins, which actually bind to water and then move the water into the cell. And that's why roots are so good at absorbing water, and they have a transport protein to bring the water into the cell. And the aquaporin is called the name of the transport protein. You probably don't need to know the name of the transport protein that moves water into the cell. The point is that this cell membrane is fluid, and the molecules, unless they're anchored in place like the peripheral proteins, they are free to move about in the cell membrane. The cell membrane shows selective permeability. This means the cell membrane allows some molecules to enter the cell or leave the cell, but not others. No large molecule can get through the cell membrane unless it has a transport protein. And that is, a large molecule is just too large to move across the lipid bilayer of the cell. So if the cell wants that large molecule to come into the cell, it has to make a transport protein to bring that molecule into the cell. Any question about any of that? And one large molecule which actually does move into the cell this way is DNA. If the cell takes up DNA, it must have a transport protein to bring the DNA into the into the cell. Ions either do not move across the lipid bilayer or they move very, very slowly. So ions cannot generally move across the cell membrane. Lipids can move right through the cell membrane. Lipids are obviously a lipid-soluble molecule, and the fatty acid portion of the lipid bilayer of the cell membrane is lipid, and so a lipid can move right into the cell. Realize that there are not too many free-floating lipids in the blood. There are some, but most of the blood is water and water-soluble molecules. But if a lipid were to come into the cell, it would get right through the cell membrane. Smaller molecules can also move right through the cell membrane. The two important ones are oxygen and carbon dioxide. They can diffuse right across the cell membrane. Oxygen moves into the cell by diffusing into the cell. Carbon dioxide tends to diffuse out of the cell. Water is another molecule which I mentioned is small enough that it can move through the cell membrane, but it has to do it in a special way. And I kind of said it right here. So there I did it. I don't remember where I did it. And what happens is the water waits for this phospholipid to move out, and then the water can take this position and then has to wait until this phospholipid moves out of the way, and then water can move into this position and then into the cell. So water does not move into the cell that readily, but it can move into the cell. And for like roots, which want the water to move very rapidly in the cell, we have transport proteins to move the water into the cell. Any question about any of this? So the cell membrane allows selective permeability, allowing some molecules to move into the cell, and other molecules cannot move into the cell. Now, if the cell wants the molecule and the molecule cannot move across the cell membrane, then the molecule will make a transport protein. And that would be actually all amino acids or and all carbohydrates. If the cell wants it, it'll make a transport protein. Carbohydrate would be something like a simple sugar. Cannot move through the lipid bilayer unless it has a transport protein. All right, let's talk about some miscellaneous aspects of the cell membrane, also called the plasma membrane. The plasma membrane of bacteria have the enzymes for ATP production. This is not true of eukaryote cells, but of bacteria, the majority ATP is made in the cell membrane, and they have the enzymes for making ATP in the cell membrane. If the prokaryote cell is photosynthetic, the photosynthetic pigments are found in the foldings of the cell membrane. Somebody's doing chat here. Let's see what's going on.
Oh, that was just me talking to the
Right.
classmates.
The photosynthetic pigments are found in the foldings of the cell membrane called chromatophores or thylakoids. Let me blow this up. I can't blow it up further. Why is that? Oh, there it goes. For some reason, it wouldn't allow me to do it. So here we have the cell membrane, and it comes like this, and then has a fold, and then it goes like that, and there's another fold here. And then we're not seeing it here, but there's a fold here, and then there's the next fold here. So this is actually coming around here, but we're not seeing that one. And then there it goes there, and there's its mother fold. And there's the folds in the plasma membrane, which contain the photosynthetic pigments. The plasma membrane can be damaged by alcohols, quaternary ammonium compounds, and then the polymyxin antibiotics like polymyxin B. How it damages cells is by putting holes in either the cell membrane or the outer membrane of a gram-negative cell. And then alcohol and quaternary ammonium compounds can dissolve the cell membrane. If you have a hole in the cell membrane or dissolve the cell membrane, the molecules of the cytoplasm can then leak out of the cell, and that would kill the cell. Any question about how we damage the cell membrane? Let me shrink that down now.
No, but I do have a question that's slightly off topic, real quick.
Uh-huh.
The for the study groups, you know how you wanted us to submit our weekly increments,
Uh-huh.
like what we studied, when we studied, etc.
Uh-huh.
What file did you want us to put that under again? Which tab?
It's the extra credit study group for that week.
Okay, so it's individually every
Yeah, you have
week.
to do it each week,
Okay.
and
Okay, thank
everybody
you.
should be doing it, although one person can make the document, send it, email it to the other students, and then you can all submit the document. It doesn't matter if you each do it or one person does it. But the point is, do submit it if you want the extra credit.
Okay, and is the schedule one every week as well? Do we submit them both, or was that just for the first couple of weeks?
I didn't understand that question, so why don't you discuss that during the lab or something? Because this is
Sure.
taking too much time, okay?
Yeah, no
Okay,
problem. Thank
but like I said, I didn't quite understand that, so I'm not sure how to answer. So,
you. No worries. I'll ask later. That's
all
fine.
right. All right, so movement across the membranes, uh, movement of materials across the plasma membrane is critical to cells. There are two processes that molecules can move to bring the molecule across the cell membrane. There's a passive process and an active process. A passive process, the molecule moves down its concentration gradient, moving from an area of higher concentration to an area of lower concentration. This costs the cell no energy to move the molecule across the cell membrane. And then there's an active process where the molecule moves against its concentration gradient, meaning the molecule is moving from areas of low concentration to areas of high concentration. This requires the cell to spend energy. And it also requires a transport protein. For each molecule moved across the cell membrane, the cell has to use one ATP molecule to move one molecule across the cell membrane. So let's say we want glucose to be moved into the cell, and the glucose is lower concentration outside the cell than inside the cell. The cell will make an active transport protein, which will bind to glucose. And for each glucose pumped into the cell against its concentration gradient, the cell will expend one ATP molecule to move that molecule of glucose against its concentration gradient. And that's the active process. The passive process, the molecule just moves down its concentration gradient. The cell needs no energy expense to move the molecule. And there's three ways that a passive process can happen: by simple diffusion, where a solid simply moves down the concentration gradient; by facilitated diffusion, which is once again a solid moving down its concentration gradient, but the molecule moves across the cell membrane by a protein. And then there's osmosis, where the water moves passively down a concentration gradient. Water is not a solute, it is a solvent. And the movement of water is so important to cells, we give it the term osmosis. Any question about any of that? All right, so let's talk about passive process, simple diffusion, the movement of a solid from areas of high concentration to areas of low concentration. Simple diffusion can happen with a membrane, but it doesn't have to happen with a membrane. Like when we put a drop of dye in the beaker here, the water will slowly disperse in the beaker of water by simple diffusion, where the dye will move from areas of high concentration right here to areas of low concentration throughout the rest of the beaker of water. And given enough time, excuse me, the dye will move by simple diffusion, so the dye will be equally dispersed throughout the water and will be a very light purple color everywhere in the water. There will be no dark purple. Now, simple diffusion across the cell membrane happens with oxygen and carbon dioxide, where the molecule simply moves from areas of high concentration to areas of low concentration. So oxygen moves from outside the cell by simple diffusion to inside the cell, because the cell has less oxygen than the outside the cell. And then carbon dioxide is higher inside the cell than outside the cell. So carbon dioxide moves across the cell membrane by simple diffusion because it's simply moving down its concentration gradient. Any question about simple diffusion? Facilitated diffusion always requires a transporter protein. It's a protein that facilitates the diffusion of the molecule across the cell membrane. I'm calling it a transporter protein, or you can call it a channel protein, because this molecule does not, or the protein does not require the use of energy to function. It simply allows the molecule to move from areas of high concentration to areas of low concentration. This is saying glucose, but glucose isn't a good example. A better example is fructose. The only way fructose moves into your cells is by facilitated diffusion. Fructose, like when you're eating a bunch of fruit, in your gut you'll have a lot of fructose. And the fructose in your gut will be higher outside the gut cell than inside the cell. And the fructose will move through this transporter protein or the channel protein into the cell. But once the fructose outside the cell equals the fructose inside the cell, the movement of fructose will not move into the cell any further. There'll be no net movement of fructose into the cell. And that means fructose, you'll never digest all of it because the digestion will stop once the concentration outside the cell equals the concentration inside the cell. And that's good to know. If you're eating a bunch of fruit, you'll never digest all of the fructose, because fructose moves into the cell by facilitated diffusion. With glucose, when the glucose is higher outside the cell than inside the cell, it will move into the cell by facilitated diffusion. And the glucose will move down the concentration gradient from areas of high concentration to areas of low concentration. The difference with glucose is once the glucose equals the concentration outside the cell equals the concentration inside the cell, this protein will change from a channel protein or a transporter protein to a transport protein. And then the transport protein will move or transport all of the glucose outside the cell to inside the cell. And that means glucose, if you eat it, like glucose breaking down sucrose into glucose and whatever the other sugar is, like it's fructose, and all of that glucose will be transported into the cell. And that's a little different where glucose initially uses facilitated diffusion when the glucose is higher outside the cell than inside the cell. But then this protein changes, and then it changes into a transport protein. And then all the glucose outside the cell will be transported into the cell. Any question about that? And that's why glucose isn't a good example of facilitated diffusion, but fructose is. All right. So both simple and facilitated fusion solutes are transported down their concentration gradient, moving from areas of high concentration to areas of low concentration. Simple diffusion can occur with a membrane, but it doesn't have to have a membrane. Facilitated diffusion always happens with a membrane. And there always has to be a channel protein to facilitate the diffusion of the molecule. Osmosis is the movement of water down its concentration gradient, but water is not a solute, it is a solvent. And as water moves down its concentration gradient into the cell across the plasma membrane, we call this osmosis, where water moves from areas of high concentration to areas of low concentration. So the net movement of water into the cell occurs by osmosis. And then I guess again, if your cell is in an area where there's less water outside the cell than inside the cell will lose water by osmosis. So water moving across the cell membrane by osmosis. Osmosis always is the movement only of water or a solvent across a selectively permeable membrane where water moves down the concentration gradient. Realize that where water is higher, the solute concentration is lower. So when the water is higher, the concentration is low, the solute concentration is low, and the water moves to the areas where the solute concentration is higher. The tonicity of water is not defined by the concentration of water, it's defined by the concentration of the solute. And we have three kinds of osmotic solutions, which is shown here. Did somebody have a question?
I just wanted to I was trying to remember this. So water is a solvent because it dissolves solutes, right? That's why it's not a solute, is because it's
a solute is something that dissolves in a solvent.
And so water would be the
solvent is something that dissolves other solutes. But water itself is not a solute, it is a solvent.
Okay.
Okay. All right. Anyways, there are three types of tonicity: isotonic, hypotonic, and hypertonic. So the tonicity is defined by the concentration of the solute. And these are relative terms where it's correct to say that the solution outside the cell is hypertonic relative to the solution inside the cell. Isotonic is the easiest to explain. That's where the concentration of the solute outside the cell is equal to the concentration of solute inside the cell. When the solute is equal inside and outside the cell, that means there's no net movement of water into or out of the cell. The cell is happy because the cell is in isotonic solution. There will be no loss of water. There will be no gain of water. By no net movement of water, we're not saying that water never moves into the cell, but if a water molecule does move into the cell, one water molecule moves out of the cell. And that means there's no net movement of water into the cell in an isotonic solution. Any question about isotonic? I guess what we should do is go ahead and blow that up so you can see it better. A hypotonic solution is where the solute concentration outside the cell is lower than the solute concentration inside the cell. In the case of a hypotonic solution, water moves into the cell because water moves down a concentration gradient, and when the solute concentration is lower outside the cell, that means the water concentration outside the cell is higher. And so water moves down the concentration gradient, moving into the cell. That will cause the cell to swell. The cell membrane will expand until the cell membrane will pop. If the cell doesn't have a cell wall, the cell membrane will expand until it bursts. And maybe you saw that in biology. Oh, what the heck is that? Biology 160 or 161, where you take red blood cells, you put it into a beaker of water, the red blood cells will expand until they eventually pop, and then you lose the red in the beaker of water as the cells burst. Because the red blood cells were put into a beaker of water in a hypotonic solution. If the cell has no cell wall or the cell wall is damaged, a hypotonic solution is dangerous for the cell because the cell will burst by osmotic lysis. Most bacteria live in a hypotonic solution. Like when you get a bacterial infection in your tissue, the bacteria is in a hypotonic solution. But the bacteria have a cell wall, and the cell wall will protect the cell membrane because the cell membrane will expand, but will only expand up to the cell wall. And then the cell membrane cannot expand any further. If the cell membrane cannot expand further, there's no more room for water outside the cell to move inside the cell. So if water tries to move in, there's no room for it, so it cannot move inside the cell. And that will protect the cell from osmotic lysis. And that's what the cell wall does, protect the bacteria cell from osmotic lysis. And that's why penicillin G tends to damage bacteria cells when they have an infection in your tissue. And that is the penicillin G puts holes in the cell wall by preventing the polypeptide linking of the cell wall. And then the cell membrane will expand out of that hole in the cell wall. And that's how the penicillin G will damage the cell, because the water will continue moving into the cell until the cell membrane bursts out of the hole in the cell wall. Any question about any of that? A hypertonic solution is where the solute concentration outside the cell is higher than the solute concentration inside the cell. That means water will leave the cell, moving down its concentration gradient, because there's less water outside the cell than inside the cell. that will cause water to leave the cell, leading to shrinkage of the plasma membrane, where you can actually see the plasma membrane pull away from the cell wall. That will lead to plasmolysis as the water leaves the cell. That generally leads to cell death. Most bacteria cannot live in a hypertonic solution for very long, like E. coli. I'm not sure how long, but after a day in a hypertonic solution, the E. coli cell will die by plasmolysis. If the bacteria, on the other hand, has something to prevent the water from leaving the cell, like Mycobacterium has mycolic acid in the cell wall that prevents the water from leaving the cell can survive in a hypertonic solution for much longer than a day. Or if the cell has an endospore, the cell can live in a hypertonic solution for a very long time, depending on how good the endospore membrane is. But the cell can survive with an endospore, depending on how good the endospore is. It could survive for many, many, many years, even millennia. And how they know is they've obtained endospores in the deserts of Egypt, like in a mummy, and the endospore, once they take it out and add water, the endospore germinated even after more than 8,000 years. Any question about plasmolysis and cells in a hypertonic solution? This is actually why molasses can be stored at room temperature for long periods of time. Could be years. My mother had a bottle of molasses that looked good. We never tested it, but we figured it was at least 30 years old. And that is the bacteria falling into the molasses underwent plasmolysis, where the water left the bacteria, and that either killed the bacteria or put them in stasis, like if the bacteria had an endospore. I'm not sure how long mycobacteria could live in a hypertonic solution. It's a long time, but I don't know if it's 30 years or more.
I had a question.
Uh-huh.
Is that the same reason why like honey lasts so long?
Yes. Honey also is a solution that has a high concentration of solute outside the cell, which is the fructose and the glucose in the honey mainly. And that's what makes the honey so thick. And a cell falls into it, it's in a hypertonic solution. The cell will lose the water and undergo plasmolysis. My mother's had a honey for at least a year, and it usually crystallized by that time. But so honey can last a long time too. It does not need refrigeration. In fact, you don't want to refrigerate honey because that will form the crystals in the honey. All right. The movement by positive processes is okay when the concentration of substance required by the cell is higher outside the cell than inside the cell. But what about a cell in an environment with a low nutrient concentration, such as a cell in the open ocean? The cell is much more likely to have the molecules it needs inside the cell than it'll find molecules outside the cell. Or a cell in the desert. It's much more likely to find the molecules it wants inside the cell than outside the cell. And so what's the cell do? It can't use a passive process, but it could use an active process, moving the molecule against its concentration gradient by using a transport protein. So in such environments, the cell can accumulate nutrients against their concentration gradient by using active transport. This requires the energy to move the molecule against its concentration gradient, because the cell needs to perform work, moving the molecule against its concentration gradient. And how it does that, it uses an active transport protein to pump the solute molecule against its concentration gradient. For each molecule moved into the cell through the transport protein, the cell has to use up one ATP. The ATP is broken down into ATP, and then that energy is used to transport the molecule against its concentration gradient. The transport protein, remember, is generally molecule specific. One specific transport protein moves one specific molecule against the through the cell membrane. For example, we have a transport protein which can move glucose, and we have another transport protein which can move lactose, the sugar lactose. Any question about any of that? All right. So in eukaryotes, the only way we can move molecules against their against their concentration gradient is by using active transport, where we have a transport protein which pumps the molecule through the cell membrane against its concentration gradient. Usually that's into the cell, but if the molecule wants to get rid of the molecule, like you have a toxin inside the cell and you want to transport it outside the cell, and the toxin is higher outside the cell, you can use an active transport protein to move the molecule out of the cell. But for most of the molecules we're going to talk about, the molecules the cell will want, glucose, fructose, galactose, amino acids, will have the active transport protein which moves it into the cell. Anyways, that's the only way a eukaryote cell can move it into the cell. Prokaryotes have another way. They can move the molecule into the cell by active transport, but they can also move the molecule into the cell by group translocation. Just checking to make sure I have enough time to talk about group translocation. Group translocation is found only in prokaryotes. It is a special form of active transport where a molecule is moved across the cell membrane by a transport protein. So you do have the transport protein. It does require the cell to expend energy, but that energy does not usually come from ATP. It comes from some high energy phosphate compound, such as PEP. You don't really need to know about PEP. And when the molecule is moved through the protein, the molecule is chemically altered. So glucose moving into the cell by group translocation moves against its concentration gradient. The cell does need to spend energy to move the molecule against its concentration gradient. And there is a transport protein, and the transport protein pumps the molecule into the cell, but while the transport protein is moving the molecule into the cell, it chemically alters the molecule. So glucose moving into the cell will not be moved into the cell as glucose. It will be glucose outside the cell, and then when it moves into the cell, it'll be chemically altered by adding a phosphate to the glucose. And so you'll have glucose 6-phosphate inside the cell. It's thought that this alteration helps prevent the escape of the molecule from the cell, and then allows the cell to accumulate substances more easily inside the cell. Let me explain that a little better. Here we have a cell. And let's say we have let's say we have 10 molecules of glucose already in the cell. And we have 5 glucose outside the cell. Now, if you were to move all 5 of the glucose inside the cell, you would, at the very last, you would be pumping this 1 glucose. Let's go 1 here. Will be pumped into the cell against 14 glucose inside the cell. So the concentration gradient is 1 outside, 14 inside. So that concentration gradient is very steep. Pumping 1 against 14, assuming they're all moved by active transport. So this is by active transport. I guess I should say that. Oh, blow that up. But if you move half of them by active transport and then half of them by group translocation, when we're moving the last one, instead of being 1 against 14, it'll now be 1 against 12. So this is by group translocation. Excuse me. A little bigger. And a little bigger. So it doesn't matter which we do because it'll be both active transport and group. Oops. And group translocation. You'll be pumping 1 against 12. Now 1 against 12 is still pretty high, but it's less steep than 1 against 14. And so if you use both active transport and group translocation, it's easier to move the molecule inside the cell because the concentration gradient is not as steep. Any question about any of that? All right, let's leave that there just in case. And then let's go ahead and detail this a little bit. And that is by group translocation, you're going to eventually move all of them outside the cell and inside the cell. Half of them will be, actually, half of them would be 12 glucose inside by active transport. And then the other inside would be, let's go ahead and just make it 3, moved in by group translocation. And this isn't glucose. Bigger here. They're going to be 3 glucose 6. I don't know how to do that. Phosphate. Yep, that's equal. It should be not an equal, but a dash. So 3 glucose moved in, or 2 glucose moved, 2.5 exactly, moved in by active transport, and then 3 moved in by group translocation. And that means we'll have something like 12 glucose inside the cell and 3 glucose 6-phosphate moved inside the cell. So actually the one moving by group translocation isn't 1 to 12. It's going to be pumped, it's actually pumping 1 against 2. Because earlier we only had 2 glucose 6-phosphate into the cell, and that's a much lower gradient than 1 against 14. Everybody hopefully sees that. And so that would be much easier to pump it into the cell. Anyways, group translocation only happens in bacteria and archaebacteria. It does not happen in eukaryotes. Any question about any of that? All right, let's move on. The cytoplasm is the substance inside the cell membrane. It's mostly water, so about 80% of the cytoplasm is water. It does have dissolved solutes in it, like obviously glucose and all the amino acids, all the other molecules inside the cell. Inside the cytoplasm, we do have the nucleoid region. That's wherever the DNA is dissolved in the cytoplasm. There is no distinction between the nucleoid region and the cytoplasm, meaning there's no division. It's wherever the DNA tends to float in the cytoplasm. We also have ribosomes and inclusions. These are so big that they do not go into solution. Blow that up a little bit. Inclusions are molecules that accumulate in the cell, and there's so many of them, they precipitate out of solution. And then you have this like clump inside the cytoplasm. Ribosomes are so big, they do not go into solution. And there's a ribosome there. That would probably be an inclusion. And then there's a little dot there. That's actually a ribosome. Any question about any of that? Any questions about anything? If not, I think this is a good place to stop. So I'm going to stop here, and I'll see you at 7 for the lab. All right, no questions. I'm going to say goodbye.